I am a consultant in medical oncology and I work at Adam Brooks in Cambridge. And I spend 50% of my time working for the NHS, where I treat kidney cancer and melanoma. And the other 50% of my time I spend working for the University of Cambridge and Cancer Research UK, where I co-lead the Urological malignancy programme, and I run a research programme focused around improving treatments for kidney cancer. I suppose the things that initially sort of caught my attention was the fact it was the Milner, and knew the Milner for their sort of reputation. The thing that attracted me to this particular call was that it was a sort of rapid turnaround pump priming grant for sort of data sets that were particularly multiomics data sets. For data that was ready to go, and we had just that we were really looking for somebody to have a really in-depth look at. So it suited our needs really well, actually, at the time. The proposal was around a clinical trial that we ran. It was led by Cambridge researchers, by Professor Grant Stewart in the Department of Surgery, also my co-lead in the Urological malignancy programme. And that clinical trial was a phase 2 clinical trial. It was run across the UK in a number of different centres, and it tested a drug called axitinib, which targets blood vessels in kidney cancer. And the aim of that trial was to try to shrink down thrombus, which is blood clot, and tumour clot associated with the kidney cancer. And we'd finished the trial, and we'd actually done most of the analysis that was attached to that, and we had lots of multiomics data sets. So we had DNA, RNA, we had protein. And what we really wanted was to look at, could we identify factors within the patients and within those data sets that might be able to tell us who's likely to respond to the axitinib treatment and who isn't, because we found that about 40% of people did have a response in terms of the cancer and the thrombus associated with it shrinking, but that still leaves a large proportion of patients who didn't respond to it, and we really wanted to find out how we would identify those patients, and actually could we identify what other drugs perhaps we wanted to pair with that axitinib to make the treatment better. The project didn't go to plan, but only in a good way. So I think on about our second meeting, I realised, oh my goodness, this is charging along. I think by about the first sort of the second meeting or so, which was about 6 weeks to sort of 2 months into the project, I realised actually we've got the data that I expected to get, and some of the answers that I expected to get, and then it only improved from there. So we got more and more in it. With every meeting, it was so exciting because, you know, you look forward to seeing what the results had shown next, and it really went beyond my expectations, actually, and it was a really brilliant experience. This project has really fitted nicely into improving treatment outcomes. How can we select patients better for treatment? And actually the results have been really excellent. And so what that's allowed us to do is now we're applying for a bigger grant, which validates the results from the sort of in silico experiments. So we'll now go forward, and we're applying for money to confirm those results in cell lines and onwards. And then what we will do is, when we identify which drugs actually are the best, is that we can apply that on our clinical trial platform. So we've got a couple of clinical trials as part of the programme that we run, and those are really, again, focused on trying to work out, are the drugs working like we think they should be in kidney cancer, and so the drugs can then go into our pipeline to actually have really quite quickly apply those results that we've got from this pump priming to patients. It's been very, very interactive, which I really enjoyed, and it's been really nice to work with, a whole range of people who've got biological expertise, you've got, therapeutics expertise, you've got the computational expertise, and it's really nice drawing all that together into a really sort of streamlined team. Overall, a really excellent experience.